Bismillahirrahmanirrahim. Assalamu alaikum students, welcome back. Today we will be discussing pairwise sequence alignment. In the previous lecture we discussed about biological databases, different types of biological databases that are there, and from today we will discuss some tools that are being used in bioinformatics, that are being used commonly in bioinformatics. And first of these many tools is the sequence alignment. And why we are discussing this in the first place is because sequence alignment lies at the heart of bioinformatics analysis. Okay, so the sequences, new biological sequences are being generated at exponential rates. As we discussed previously, that with the um, sequencing cost becoming low every day, so the number of sequences generated are getting higher and higher. So the new biological sequences are being generated, and it's important that you annotate the sequence, that what this sequence means or what this sequence codes for. Okay, so the first step or the sequence alignment is that then becomes the most fundamental process by which we can annotate a sequence. So sequence alignment, we can say that it's the first step towards structural and functional analysis of newly determined sequences, right? Uh, and this is a process, by which we compare the sequences together, and we are interested in some common character patterns. Okay, what do we mean by common character patterns? The sequence, they dictate the function of the molecule. For example, if there is a sequence of a protein that we know, so that sequence will tells us that what kind of 3D structure that protein will make, and what kind of function that protein possibly possess. So we look for common character patterns, and if we find certain similarity, or if we can figure out that our sequence is very similar to some known sequence, then it means that since sequence is the same, the function most probably will also be the same. So the nucleotides and the amino acids, they are basically the building blocks of DNA and proteins. We all know this thing, and they can be considered as molecular fossils that encode the history of millions of years of evolution. Now, what do we mean by this? Uh, these molecules, they can be considered as molecular fossils because they still have the traces of evolution, and because some residues, they perform key functional and structural roles, and they are preserved by natural selection. This can further be we can understand with this a simple figure, that we have a DNA or a protein sequence, and during the course of evolution, certain mutations, they will be accumulated. Okay, so these mutations, they are random mutations, and some of these mutations, they will be selected, or they will be uh, um, accepted by natural selection or during the course of evolution, and some of the mutations will be lethal for the species or for the organisms, and these kind of mutations, they will be rejected straight away, because mutations in uh, those sequences or proteins that are critical for survival of the cell, uh, these mutations will not be accepted by the evolution, and they will be straight away rejected. Okay, so with the accumulation of these mutations, the sequences, they will start to diverge. And they might get some uh, new functions over the course of evolution. Okay, so before going deep into this, let's so we'll try to grasp this concept. So we'll try to understand this concept by looking at the evolution of a telephone. Okay, so you might have seen these telephones, you might have used it, or you might not have used these older version of telephones. But the older telephones, they were quite big, and their function was very was different. But now we have smartphones. They are the size of a palm, and they have tons of functions. You can do a lot of productive stuff using your telephone mobile phones. But the basic function of all these devices is to make a call and to receive a call. So when you are low on battery and you turn on the battery saver on, so the only function that will be available on your mobile phone would be making call or receive a call. Okay, so this is the basic function of a telephone that stayed there for all of this time. But with the passage of time, obviously the new functions were introduced in the phone to make it look better, to make these devices more productive. Okay, so similarly, when we come to the DNA and proteins, so suppose there is a protein in some common ancestor, and and that protein performs some function, and due to these random mutations that have been accepted by natural selection, and the mutations that they were accumulated, so the current day protein might be different at the sequence level, but they will be performing more or less the same function because the protein was derived from a common ancestor. So the function of protein, the basic function of the protein might remain the same, but it may happen that because of accumulation of these random mutations, protein might get some additional functions, or protein might get more stable, or they become resistant to proteases, and so on and so forth. Okay, so this is what's happening. That is why we are saying that these uh, molecules like DNA and proteins, they are the molecular fossils, and so this is the concept. So before moving towards the tools for sequence alignment, I just want you to uh, make you know clear the concept of sequence homology, sequence similarity, and sequence identity. So these are some terms that are being used that you might read somewhere, or you read some papers. So sometimes these terms, they are being used interchangeably, but there's a slight difference. I mean, each term is distinct and each term has its own meaning. For example, if we talk about sequence homology, so sequence homology is when two sequences, they share a common evolutionary origin, then they are said to have homologous relationship, or they are said to share homology. Okay, so if two sequences or two molecules, they have a common ancestor, they are said to be homologs. Okay, so sequence homology is more of a qualitative term, which is basically a conclusion about common ancestry based on the sequence similarity. Okay, so what do we mean by uh, qualitative term? Because suppose there are two sequences, they can be either homologous or non-homologous. We cannot say that two sequences, they are 40% homologous, or you cannot say that these two sequences are 70% homologous, right? So either the sequences are homologous or they are non-homologous. Just like we can say that two persons, they can be brothers or they are not brothers, right? The next term is the sequence similarity. Sequence similarity is more of a quantitative thing, which is the percentage of aligned residues that are similar in physiochemical properties. Now we'll discuss shortly about these. What do we mean by similar in physiochemical properties? But the sequence similarity is basically the percentage of aligned residues. So when you align two sequences together, the number of sequences that are aligned with each other, that is the sequence similarity. So we can say that the sequence is 40% similar, or 70% similar, or 80% similar, right? So here the quantitation is there. You can quantify the the extent of similarity. And sequence identity is same as sequence similarity if you are working with DNA, because DNA, they are just four nucleotides: adenine, guanine, cytosines, and thymines. So for DNA, sequence similarity and sequence identity, they can be used interchangeably, but for protein sequences, sequence identity and sequence similarity has different meaning. So if you are considering, or if you are studying protein sequences, sequence similarity means the percentage of aligned residues that are similar in physiochemical properties. Now what do we mean by this? There are 20 different amino acids, and amino acids are further grouped into, like, there are some amino acids that are acidic, some of them are basic, some of them are polar, some are non-polar, some contain the aromatic ring with it. So they are classified into different groups. So if one amino acid is changed into another amino acid, but both these amino acids belong to the same group. For example, an acidic amino acid was changed into another amino acid which is an acidic amino acid, then the physiochemical properties will remain the same, and the protein most likely will retain its functions, and there will be no disruption in the function of the protein. Okay, so this is called as sequence similarity. And if we talk about sequence identity, it means that the amino acids at certain position, they are identical. So if we have a leucine in one sequence, at the same position we have a leucine in the other sequence. So here the sequence identity and sequence similarity has somewhat different meaning. Now the question arises, at what similarity level can one infer homologous relationship? Means how much sequence identity or sequence similarity should be there for us to say that these two sequences, they are homologous to each other. Okay, so that depends upon two major factors. The first is the type of sequence, and by the type of sequence we mean that whether it's a sequence of DNA or it's a sequence of a protein. And the second factor is the length of the sequence, right? uh, that there are just 4 nucleotides in DNA. Okay, so there is a probability, or there is a chance that 25% chance that an identical or similar residue is there because of chance. Okay, and we have 20 amino acids in proteins, so there is a 5% chance that you will get a residue that is identical at a specific position randomly by chance. Okay, so keeping in view uh, these stats, we can further try to understand this with the help of this example. You can see this graph, that there is a sequence identity at the y-axis and there is a sequence length at the x-axis, and there are three different zones. There is a safe zone, there is a twilight zone, and there is a midnight zone. So safe zone is the zone where we can safely say that the two sequences, they are homologous. Twilight zone is the zone in which we are not confident about the homologous relationship. We cannot say for sure that the two sequences are homologous or not. But if the sequence homology is less than 20%, identity is less than 20%, we can be sure that these two sequences that we are comparing with each other, they are not homologous, right? So for example, in this case, suppose there is a sequence that is 100 residues long. So for 100 residues long, if sequence identity is 30% or more, we can safely say that the two sequences, they are homologous to each other. And if the sequence identity is between 20 to 30%, we are not sure whether to say that these two sequences, they are homologous or not. But we will be sure that less than 20% identity is not enough for inferring homologous relationship. Okay, so with this graph we can see that if we increase the sequence length, we can decrease the threshold of sequence identity. But if the sequence length is less, like sequence length is of 50 residues, then sequence identity threshold of sequence identity should be more. So this is how we can infer homologous relationship. Next, uh, we'll now move towards the methods of uh, methods that are being used for uh, aligning sequences. But first, uh, let's make it clear that the goal of sequence alignment is to find the best possible alignment between two sequences. Okay, so this is our goal, that we want to find the best possible alignment between two sequences. Okay, so we have two different methods that we will discuss in this course. One is the dot plot method. Dot plot method. Dot plot method is a visual representation of two sequences. Like we have a matrix with x-axis and y-axis. We write one sequence on the x-axis and the other sequence on the y-axis, and then we compare each residue of one sequence with every residue of the other sequence. But the results will be inferred by visual representation. Now we will see when we'll do some examples for this. And the other method is called dynamic programming method. We'll discuss in detail what's the difference between dynamic programming and dot plot method, and the two types of alignments that are being generated. uh, One is the local alignment and the other one is the global alignment. Now let's discuss what do we mean by these. So what is the dynamic programming? By dynamic programming we mean that we are breaking down a larger problem into smaller sub problems and then solve each sub problem in order to solve the bigger problem. Like we have a bigger problem, we break down that bigger problem into smaller sub problems, and each sub problem is then solved, and at the end we'll compile all the data together, and that big problem is being solved by this way. So it is a computational method to find the best possible best optimal alignment between two sequences, and this method compares every character in the two sequences and generates an alignment. Okay, so this will become more clear when we will do the examples, right? And what's the difference between global and local alignment that we just discussed? Global alignment is when the two sequences, they are of approximately the same length, and when the two sequences, they are closely related to each other, then a global alignment is generated. So in global alignment, sequences are aligned end to end. So both the if you have two sequences, they will be aligned from one end to another, and this method is good for sequences that are similar and they are of the same length. As for the local alignment, they align stretches of sequences with the highest density of matches. Means that we do not align sequences end to end. We try to find uh, sub regions, I mean the region within the sequence that has a highest density of matches, and it generate more than one aligned regions. I mean by local alignment we will get more than one alignments, because there can be possibly more than one alignments. And this local alignment is good for aligning sequences that are similar at only some positions and different at others. So if the two sequences, they are they are not closely related and divergent sequences, then for these two sequences opting local alignment method would be a better option, because we will be then more interested in the regions that are functionally important. And these local alignment method is good for sequences that vary in length and that share a conserved domain or motif. Okay, so all these things will become more clear um, later when we will be doing some exercises for this. So that's it for today. In the next lectures we will discuss a dot plot method of aligning two sequences, and we'll also discuss how we can generate a global alignment using Needleman's Wunsch algorithm. And then we'll also discuss about local alignment, where we will discuss how Smith-Waterman algorithm is used or employed for generating local alignments. uh, we will have question answers related to this topic in our next QA session. Till then, Allah Hafiz.